Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, so you know CRISPR-Cas9, right? The incredible DNA editor.
Yeah, it's pretty much a household name in biology now.
Totally. But what if we told you the future of RNA editing comes from somewhere? Well, way more ancient and kind of unexpected.
Like, imagine a tiny bacterial defense system, just a simple toxin protein that originally just stopped bacteria from growing.
Right, basic stuff.
But then, over millions of years, it evolves. It turns into this programmable, super precise RNA editor.
It sounds like science fiction, honestly. But it's real. It's the discovery of this new CRISPR-like system called Cas13. Welcome to the deep dive. This is where we unpack the big insights from complex research, try to turn that dense info into something fascinating and hopefully digestible.
And today we are diving into a really groundbreaking paper from Cell. It basically rewrites a chapter of CRISPR evolution.
Seriously.
Yeah, and it opens up some incredible new possibilities for molecular medicine. We've waded through all the technical jargon and methods.
All the figures and supplementary data. Exactly, to bring you the core aha moments. You can walk away feeling like you really get it without, your brain melting.
Right. So our mission today is to explore these unexpected evolutionary roots of RNA targeting CRISPR. We're focusing on how this compact bacterial toxin-antitoxin pair.
That's TA system.
Right, this TA system got repurposed by evolution, how it became a precise, reprogrammable tool that cuts RNA.
And we'll show you how this discovery could genuinely transform things, diagnostics, maybe even gene therapy down the line.
It's big stuff.
So let's set the scene a bit. CRISPR-Cas, like we said, it's changed the game for DNA editing. Snip, paste, incredible precision.
Absolutely revolutionary.
But RNA, that crucial messenger molecule, the tools for targeting it.
Yeah.
Well, they're still a bit scarce, aren't they? Or at least they have issues.
They do. Robust RNA tools are harder to come by. The main ones we have, these Cas13 effectors, they're useful, sure, for knocking down RNA levels, but they have pretty significant drawbacks. The big one is collateral cleavage.
Ah, yes, the molecular chainsaw massacre, I think I called it earlier.
Yeah, that's not a bad way to put it. You aim it at one specific RNA transcript.
And it just starts chopping up other stuff nearby.
Exactly. Unintended RNAs get cut too. That really limits the specificity, which is, a massive problem if you're thinking therapeutically. You cannot have those off-target effects.
No way. Precision is everything there. And it's not just the messiness, is it? It's also their size.
Right. They're big.
Yeah.
Typical Cas13 proteins are, around 1,200 amino acids long?
That's chunky for a protein.
It is. And that makes them really difficult to deliver into cells, especially using the common delivery trucks like AAVs, those viruses we use for gene therapy. They have limited cargo space.
So too big, too messy, that's the gap researchers have been trying to fill, right?
Right.
A smaller, cleaner RNA editor?
Precisely. And that leads to this unlikely hypothesis scientists had for a while.
Which was?
They suspected that bacteria and archaea prokaryotes might have these really compact natural RNA cutting systems, these toxin-antitoxin pairs, new TA systems.
Okay, the bacterial self-destruct and antidote thing.
Exactly. So the question was, could these ancient RNA cutters, these RNases, have somehow evolved to be guided, like CRISPR? Could they learn to use a guide RNA to find a specific target?
That feels like a huge evolutionary leap. Connecting a simple toxin system to sophisticated programmable CRISPR, how do you even start looking for that link?
Yeah, it's detective work.
Yeah.
They needed to trace the family tree, basically. And they used this really clever method, a hybrid sequence and structure search.
Hybrid, meaning?
Meaning they didn't just compare the protein sequences, the amino acid chains. They also used tools like AlphaFold 2, which is amazing at predicting 3D protein structures, and DLI to compare those structures.
Ah, so comparing not just the blueprints, but the actual finished buildings, sort of.
Exactly, because structure often stays more similar over long evolutionary times than sequence does. It helps you find those really distant relatives.
Okay, so they run this massive comparison, and what tops out?
Something astonishing. Cas13, the RNA targeter we know, likely evolved from much, much smaller proteins, primarily one called AbiF,
AbiF, what's that stand for?
abortive infection F, and some related ones like F13A12 and this newly classified one Cas13. This AbiF is tiny, like 432 amino acids. Super compact.
Wow, compared to 1,200, that's a huge difference. So this was like finding a whole new branch on the CRISPR family tree.
Totally new branch. And the structural clues were just, well, undeniable. Even though they look different on the surface, Cas13s and AbiF share this unique structural feature.
A molecular fingerprint?
Kind of, yeah. It's called a circular permutation in their core HEPN domain. That's the bit that does the cutting. It's a specific, unusual arrangement of the protein chains in that region.
HEPN, higher eukaryotes and prokaryotes nucleotide binding.
That's the one. This weird structural twist is a really strong sign they share a common ancestor. And then they did phylogenetic analysis, mapping the evolutionary tree. And yeah, AbiF sits right there as this key intermediate on the path to Cas13.
Okay, so they found the ancestor, AbiF. That's huge. But then they needed to know, how does AbiF actually work? What's its day job in the bacteria?
Right. So they did another search, this time an RNA association search, looking for RNAs that consistently hang out with AbiF. And they found one, a statistically significant link between AbiF protein and a specific non-coding RNA. They named it AbiFAR.
ncRNA, AbiFAR. So a functional RNA, not one coding for protein. That immediately suggests it's doing something with AbiF, right?
Exactly. It points to a functional relationship.
So did they actually see this toxin-antitoxin thing happen in a lab setting?
They did. They tested it. When they put AbiF alone into bacteria, boom, growth slows right down. It's toxic.
Okay, the toxin part works.
But then, and this is the cool part, if they express the AbiFAR ncRNA with the AbiF protein, or even turned on the RNA first,
Let me guess, the toxicity disappeared.
completely abolished, gone. The AbiFAR RNA acts as a powerful antitoxin. It just shuts ADF down.
Wow. The bacteria has its own off switch hardwired. So how does the RNA molecule actually do that? How does it stop the protein?
Okay, so biochemical experiments showed AbiF can actually chop up its own AbiFAR RNA sometimes, which is interesting.
Like some Cas13s do with their guides?
A bit, yeah. But crucially, the intact AbiFAR RNA strongly inhibits AbiF's ability to cut other RNAs.
It stops the main RNase activity.
Right, and then they got the structure using cryo-EM of AbiFAR bound to AbiFAR high-resolution 3D picture.
And what did that show?
It showed the RNA, the AbiFAR, snakes its front end, the 5' end, right into the active site of the AbiF protein.
Like jamming a key in the lock.
Exactly like that, like putting a physical cork in the bottle. It blocks the enzyme's cutting machinery. AbiFAR normally cuts single-stranded RNA internally. It's an endonuclease, but the AbiFAR just plugs it up.
Okay, that's fascinating. AbiF is a toxin. AbiFAR is the antitoxin quark. But that's still just a bacterial defense system. How do we get from that to something programmable, like CRISPR? Where's the bridge?
That's the next piece. They identified this other protein, Cas13E, used to be called C13C1, as the critical link, the evolutionary intermediate.
Okay, Cas13E, what's special about it?
Well, first, it's still pretty compact, around 450 amino acids, much smaller than typical Cas13s.
Still closer to AbiFIs than the big Cas13?
Exactly. And crucially, unlike AbiF, Cas13E can bind a cRNA, a CRISPR RNA guide.
Ah, okay. There's the programmability, the guide RNA.
That's the key. And this is where Cas13E just blew them away. It showed RNA-guided RNA cutting activity, just like other Cas13s.
There's a but, right.
With no collateral activity when the target match was perfect.
No collateral.
Yeah.
Seriously, that's the holy grail for RNA editing, isn't it?
It's huge. Imagine precision targeting without the chainsaw effect. Just hitting the one RNA you want to hit. That changes everything for potential therapies where off-target effects were the deal breaker.
Absolutely. It opens up diseases we couldn't safely target before.
And there's more. Cas13E showed this really interesting behavior. On its own, without a guide RNA, the APO form, it actually had a little bit of weak random RNA cutting activity.
Like tiny bit leaky.
Yeah, a little bit. But then, when you add the cRNA guide, that weak activity gets shut down, inhibited.
So the guide RNA acts like an initial safety switch?
Sort of. And then the cutting activity only gets fully switched on when the guide RNA finds its matching target RNA.
Wow, so it needs the guide and the target to really activate.
Exactly. It really shows how it's this amazing hybrid. It has traits of the TA system, the guide almost acting like a target-dependent antitoxin, and the CRISPR system being programmable. It's like a switch that needs two keys turned simultaneously.
Guide and target. That's incredibly elegant. And this system combining the TA origin and Cas function, they're calling it CASTA.
Right, Cas toxin-antitoxin reflects that amazing evolutionary history.
And its compact size plus this precision. That sounds incredibly promising for actual applications.
Immensely promising. Remember the size problem. Cas is about 60% smaller than typical Cas13s.
Which means...
It means it's much easier to package into those AAV delivery vectors. You can actually fit it in the truck now.
Okay, that overcomes a major hurdle for in vivo therapies. Did they show it works in practice?
They did. In mouse models, they delivered Cas using AAVs into the retina. And it successfully silenced disease-causing RNAs involved in abnormal blood vessel growth pathological neovascularization.
So it worked in a living animal, reducing a disease process. That's proof of principle. Huge.
Yeah, it validates the potential for precise therapeutic RNA knockdown. Think about eye diseases like macular degeneration.
Yeah.
This offers a new, maybe much cleaner approach.
So how did evolution actually build this? The paper mapped out the journey, didn't it? From simple AbiF to programmable Cas13E?
Meticulously. It looks like first the antitoxin RNA, the AbiFR, probably got longer, expanded.
Okay.
Then the AbiDef gene itself likely duplicated and maybe fused together, making a protein with 2 HEPN domains.
Getting bigger, more complex.
Right. Then, over time, the place where the antitoxin RNA bound might have shifted. New bits of protein, new helices that bind RNA got inserted.
Bolting on new functions.
Exactly. And gradually, it lost that strict dependence on the original AbiFR antitoxin. That freed it up to potentially interact with other RNAs, like cRNAs from a nearby CRISPR system.
Ah, the crossover moment.
Possibly. A really key step seems to be the insertion of a specific bit called the REC helix. That part is crucial for recognizing the pair formed by the guide RNA and the target RNA, the duplex.
So that REC helix is what confers the guide target specificity.
Seems to be a major part of it. So you get this minimal set of changes, duplication, insertions, shifting binding sites, and boom, a non-guided toxin enzyme evolves into a basic programmable RNA-guided system like Cas13.
And from Cas13, it likely continued evolving, adding more domains, becoming the larger, more complex Cas13s we knew about before.
That seems to be the path. It's an incredible story of molecular tinkering by evolution.
So, stepping back, what's the big picture here? This feels like more than just a new tool.
It really is.
Yeah.
It's potentially a paradigm shift for programmable RNA editing. We're no longer limited to those big collateral-prone Cas13s for precise work.
Cas's combination of small size and clean cutting makes it a prime candidate for, well, lots of things.
Definitely. Think diagnostics. You need absolute precision to detect specific disease RNAs without false positives. Cas looks ideal.
Or allele selective silencing. That's the dream, right? Silencing a faulty copy of a gene's RNA message while leaving the healthy copy untouched.
Exactly. That level of discrimination is critical for many genetic diseases. Cas's precision could potentially enable that. It opens up therapeutic avenues that were just too risky before.
So this work really seeds a whole new branch of CRISPR biology, developing these modular RNase editors.
Yeah, it expands the whole CRISPR toolbox. It's not just a genome surgeon for DNA anymore. Now it's also becoming a potential transcriptome therapist for RNA.
A transcriptome therapist, I like that. Targeting disease at the RNA level.
It's a huge leap. Now, of course, there are still challenges.
Always are.
Yeah. We need to think about potential immune responses in humans to these bacterial proteins. That's always a factor.
And ensuring the guide RNAs are super accurate, especially with complex stuff like RNA splicing happening in our cells.
Right. Those are engineering and safety hurdles we need to clear. But the fundamental discovery, this evolutionary insight, is remarkable.
It really shows how these basic, innate immune systems in bacteria
Ancient defense mechanisms
can evolve into these sophisticated adaptive systems. It's this constant arms race between microbes and viruses driving innovation.
And it makes you wonder, what other RNA-guided systems might be out there? Maybe ones not even linked to CRISPR, that evolved down similar paths from other ancient enzymes.
Yeah, there could be a whole hidden world of these things waiting to be discovered.
So we've gone from this tiny bacterial toxin, kept in check by its own RNA antidote,
all the way to a compact, precise RNA editor with real therapeutic promise, Costa.
It's just an amazing testament to how evolution can repurpose the simplest tools into something revolutionary.
It really is. So here's a thought to leave you with. Given this incredible journey, from a simple bacterial toxin to a sophisticated RNA editor, what other seemingly simple biological mechanisms, maybe things we currently overlook, might hold the keys to completely new breakthroughs?
And what other unexpected evolutionary shortcuts has nature taken leading to tools we haven't even imagined yet?
What's the next Casta waiting out there? Keep digging.